In this chapter, we're going to talk about congenital genetic and developmental disorders. A lot of times, if you're working in pediatrics, you are working with people with congenital and developmental disorders and genetic too sometimes. We'll talk about the difference between those. So, the learning objectives are to define the basic terminology, of course, to differentiate between congenital, genetic, chromosomal, developmental, and multifactorial conditions, and to talk about inheritance patterns associated with genetic conditions. I'm not going to ask you to do Punnett squares or anything like that, but just the general idea of how it works. So, understanding the fundamentals of genetic disorders and their implications for physical therapy is the focus of our chapter. So basic terminology of genetics. I know you all had biology, but a gene is the basic physical and functional unit of heredity made up of DNA, and genes provide the instructions for making proteins or regulating cellular mechanisms. So the basic tenet of molecular biology is genes code for RNA, which codes for proteins, which then goes out and does everything in the body. So basic. Alleles are different forms of the same gene with a small DNA sequence difference that can contribute to unique physical features. So, to take a very simplistic example, and it's more complicated than this, but to simplify it, like the allele for blue eyes versus the allele for brown eyes. So, if I have we have the same gene, I just happen to have the brown-eyed allele and you have the blue-eyed allele, we're going to have unique physical features. So the chromosome is the thread-like structure in the cell nucleus that contains the DNA tightly coiled around these proteins called histones. And then there are enzymes that uncoil the DNA and code for the RNA, which then goes out and makes the proteins in the ribosomes in the endoplasmic reticulum in the cell. Hopefully, all those sound familiar. As human beings, we have 23 pairs of chromosomes, so 46 chromosomes total. 22 are autosomes, and then we have a pair of sex chromosomes. Generally, XY is female and X X is female and XY is male, but there are some combinations that come up that are a little bit different. We'll talk about. So the key terms, genetic orders relate to genes, so they're inheritable. So your parent had a different gene mutation or something like that, and it gets passed on to you. Congenital disorders are present at birth, but they're not always genetic. So cerebral palsy is a good example of that. It's not genetic, it can't be passed on through the genes, but it is present at birth, and it's non-progressive, but you have it for your whole life. So genotype versus phenotype. Genotype is the actual genetic information carried by the individual. So when they sequence genes and stuff, that's what they see. They see your genotype. All cells except for the gametes of an individual have the same genotype. So your liver cell has the same genotype as your heart cell, and they have the same genotype as your skin cells. So there's the thing that I like about developmental biology is that it's mysterious. There's still tons of mysteries to be solved. And so not all genes are expressed in all cells, and that's how we get the differentiation of cells. Do we know how it works? No. We're, hopefully people are working on it right now, but it's a mystery on a lot of this stuff. So I do like that there are some mysteries left in life. Phenotype is the expression of genes. So that's the appearance of the individual's characteristics. So I'm tall, I'm short, I I have this particular connective tissue arrangement, and you have a different one. So I always say genotype is what you're dealt, and phenotype is how you deal with it. So that's just my little for remembering it. So remember, congenital disorders are present at birth. They can be inherited or developmental, but inherited disorders can be caused by single gene expression, chromosomal defect, polygenic expression. Chromosomal anomalies are usually caused by an error during meiosis, a non-disjunction or a translocation. So when The non-gamete cells divide, they just make two copies of themselves, two daughter cells, um, and they divide in mitosis. During meiosis, you make four, and each one has half the amount of DNA. And so in our gametes, we have half the amount of DNA. So then when it combines with the gamete from the other parent, you don't have too much. So too much DNA is not a good thing. But during the meiosis, stuff happens. And I always say, relax, nothing is under control because it isn't. So there are different types of gene disorders, and there's different classifications. There's a single gene disorder where you have a mutation that affects one single gene that does something important, or it probably wouldn't be named and classified. The trait is controlled by one set of alleles. Either you have the disease allele or you have the non-disease allele. It's transmitted to subsequent generations, so it is inheritable. So examples are sickle cell anemia, cystic fibrosis, Duchenne muscular dystrophy. So those are all single gene disorders. There are chromosomal disorders, which are where you have either missing chromosomes or changed chromosomes or chromosome parts. So during meiosis, part of one chromosome gets tacked onto another. That's called a translocation. So examples of chromosomal disorders are Down syndrome and Turner syndrome, and we'll talk a little more specifically about those. So obviously it's not that simple. Nothing ever is, right? So it's complex. There are complex multifactorial disorders that involve mutations in multiple genes and sometimes environmental factors that either trigger or precipitate the disease. There are tons, large numbers of disorders that are this type. They often have a familial pattern of inheritance, but just because you have the genes doesn't mean you're going to get the disorder. So with the single gene disorders and the chromosomal disorders, if you have the gene, you have the disorder for the most part. We'll talk a little more in detail about that. With chromosomal disorders, if you have that dislocation um, or translocation or part of the gene is missing, then you have the disorder. With multifactorial disorders, you have a genetic tendency towards certain types of cancer or other things, but you might not get them. So kind of interesting and complicated. So of course I love it. I love complicated stuff. So congenital conditions or sometimes developmental disorders can result from premature birth. They are present at birth or occurring very soon after birth. So many result from birth injuries during difficult labor and delivery. So I worked with a patient who had CP, and he was one of a set of twins, and his twin was born first, and then he was deprived of oxygen while his twin was being born, and so he ended up with some congenital disorders because of that. So it can be genetic or non-genetic. In that case, it was non-genetic. It can be caused by maternal conditions during pregnancy, like spina bifida is a good example of that. It can be caused by developmental problems in utero, like the mother is exposed to different environmental toxins or pathogens. So spina bifida is an example, cerebral palsy, fetal alcohol syndrome, so that's being exposed to, poisons, chemicals, or congenital heart defects are sometimes caused because something happened during a critical part of development. So there are different critical stages in development, and in neuro, we'll talk more about stages of neurological development. But if something happens during that critical stage, then something changes in the development. Genetic conditions are caused by abnormalities or changes in genes or chromosomes. Genetic conditions are always hereditary, but they may not appear at birth. There are certain diseases that are known as um, they you don't see them until you're older. So they're called delayed lethal genotypes. So I don't know if lethal is the right word, but that's what they call it. It means you don't know you have it until much later in life. So polycystic kidney disease is an example of that. It can be diagnosed through blood tests. You can do genetic testing and through saliva tests now too. And they follow specific inheritance patterns. So if your parent has it, you have it. Huntington's disease is an example of that. And there's this like big news in science. There's now a treatment for Huntington's disease. That's amazing. So, edge of seat stuff here. Marfan syndrome is a genetic condition, cystic fibrosis, and Duchenne muscular dystrophy. I want to see cures for so many of these things, or treatments at least. And a treatment for Huntington's disease, that is major news in the science world. So with pregnancy, the different terms of pregnancy are classified. The I don't know what the 6 days. This is from the book, and I don't know where that 6 days came from. So we're just going to talk about weeks. Maybe it's 37 to 38 weeks and 6 days. I don't know. But early term is 37 to 38 weeks. That means if a baby is born at 37 to 38 weeks, it's considered a premature birth. Full term is 39 to 40 weeks. Late term, if I'm sure the mom is really anxious at this point, if the baby hasn't been born, 41 weeks to 41 weeks and 6 days. I think that's what that is. And then post term is 42 plus weeks, and preterm is before 37 weeks. So usually, like in the olden days, they used to have to let people just wait until the baby was born. Now, if you're at full term, they're going to induce labor if you haven't already naturally gone into labor. But preterm birth complications, if you have a premature baby born before 37 weeks, they might have breathing difficulties because the surfactant lining in their lungs hasn't developed yet. They might have heart defects. They might have cerebral palsy. They might have learning disabilities or feeding or swallowing problems because those are the feeding and swallowing, that's neurological integration. So they might have problems with that. They might have temperature regulation issues, abnormal muscle tone, or failure to thrive where they just aren't growing. So usually babies born preterm are put in the NICU, the neonatal intensive care unit, until they are ready to be on their own. I mean, they're not going to just send them to college right after that, but send them home with their parents. I really should drink more coffee before I record these lectures. So there are single gene inheritance patterns, and we talk about them as being either autosomal or X-linked. So autosomal, it's one of the 22 autosomes, which are not the sex chromosomes. And you only need, if it's a dominant gene, an autosomal dominant disorder, you only need one abnormal gene. And usually one affected parent provides one abnormal gene. And so there's a 50-50 risk that the child is going to have disorder every time this couple with the one affected parent has a child. There's no carrier state. If you have the gene, you have the disease. In an autosomal recessive, you could have, if only one parent has the gene, none of the kids are going to have it. They might be carriers. They have a 50% chance of being a carrier, where they have the one gene. But both parents have to carry the gene in order for you to have the disorder in the next generation. And each pregnancy has a 25% risk of having the disorder, 50% risk of being a carrier, and a 25% risk of not having the disorder. So they basically, an autosomal dominant is always expressed, and an autosomal recessive is only expressed 25% of the time. So some people think of these percentages as like, oh, if you have two kids, one will have the disorder and one won't. But that's not true. It's actually a 50-50 chance every time you do it. So you could have four kids and none of them could have the disorder. It's just luck of the draw. It's the luck of the draw. So it's and same thing with autosomal recessive. It's not like you have four kids and one will have the disorder, two will be carriers, and one will not have the disorder. It's the each pregnancy has the same odds. Okay. So just an example of an autosomal dominant disorder, Marfan syndrome is a connective tissue disorder, so it affects the gene for certain types of connective tissue. It's known to be on chromosome 15, It's an autosomal dominant. It can be a spontaneous mutation or it can be inherited from the parents. It affects connective tissue formation, and it may not be diagnosed until adulthood, depending on the manifestation of the disease. The physical characteristics are tall stature with long limbs, long fingers and toes. Scoliosis is common, and sometimes you get abnormal rib cage formation. Some historians theorize that Abraham Lincoln had Marfan syndrome because he was tall, long-limbed, had the characteristics of it, but we don't know. It's too long ago. It can have some serious complications like aortic problems, aortic aneurysm, um, and it can rupture and be fatal. I've worked with two people with Marfan syndrome in my clinical career, and one of them did have an aneurysm rupture, and luckily it was not fatal. They were able to get surgery and survive. The lung can have a bulla, which are little like pockets of congealed tissue, and a spontaneous pneumothorax could happen. The what collapsed lung. You could have retinal detachment in the eyes because there's that connective tissue again, or lens dislocation. And in the heart, you could have mitral valve prolapse. There's no cure for Marfan syndrome currently, but I have hope for everything. Surgery for heart or aortic problems, definitely. Medications like beta blockers or the ARBs to keep the blood pressure and heart rate low, and eye surgery when needed. So Marfan syndrome is interesting. Interesting. Cystic fibrosis is an autosomal recessive disorder. So both parents have to be carriers. Both parents have to be heterozygous for the allele. It's a mutation on chromosome 7. Interestingly enough, the professor that I had human biochemical genetics with was the one who discovered the cystic fibrosis gene. So that's interesting. Interesting to me. So it's an autosomal recessive inheritance, meaning each pregnancy you have a 25% chance of having an affected child, 50% chance of carriers, and 25% chance of not affected by having both unaffected genes. So there's some incidence by ethnicity. It's clearly more common in white Caucasian people, but there is still occurrence in other ethnicities. And there's the statistical layout. So cystic fibrosis, it causes tenacious mucus to be excreted from the exocrine glands. The primary effects are seen in the lungs and the pancreas. In the lungs, mucus obstructs blood airflow in the bronchioles, and it can cause permanent damage to bronchial walls. And infections are common because infectious agents love thick, tenacious mucus. In the digestive system, it can meconium ileus is a condition in newborns, blockage of the ileum, the blockage of pancreatic ducts, obstruction of bile ducts and salivary glands. It can affect the reproductive tract. Sweat glands, the sweat currently has a high, or usually has a high sodium chloride content. And before they had genetic testing, they used to, the they would suspect cystic fibrosis. The mom would like kiss the baby on the forehead and they would taste salty. And then I don't know what testing they did before genetic testing, I'll be honest. But in PT treatment, a lot of times we're treating people with chest physical therapy or postural percussive drainage. They help clear the secretions. There's also some people there's a vibration vest. You put the vest on, it's attached to like a machine that vibrates your chest to help clear secretions. Other areas of PT that we're going to include for people with cystic fibrosis are breathing and assisted coughing techniques, and we will learn those next year in our special topics class when we're doing pulmonary rehab. Ventilatory muscle training, postural training, mobilization of the thorax and breathing exercises, and general cardiovascular conditioning is also helpful. So single gene X-linked inheritance patterns. So those were autosomal disorders that we just talked about, dominant and recessive. With X-linked, of course, you get an X from your mother and a Y from your father. The Y chromosome is tiny; it doesn't have much on it, and so most of the good stuff is on the X chromosome. So if you have a recessive that's on the X chromosome, a recessive disorder, it mainly affects males because they won't have a good copy of the X chromosome. Females have a spare, but males don't. So when the carrier mother and the unaffected father have a child, sons are at 50% risk and daughters are at 50% risk of being a carrier. Um, X-linked dominants are extremely rare. I will talk about one of them. Very, very rare. That means if one of your parents has it, you're going to have it. Or if your mother has it. But anyway, Duchenne muscular dystrophy is a an X-linked recessive disorder. So it's the most common type of muscular dystrophy. It's carried by mothers. It affects sons. So sons have a 50-50 risk of the disorder with a carrier mother because the carrier mother has one good X and one affected X. So the son's going to get one of those. Um, the daughters have a 50-50 risk of being a carrier. So if they get the good X from their dad and the affected X from their mom, they're a carrier. It's possible to have spontaneous mutations, but there's no way to tell that that happened. I mean, I guess you could do a genetic screen on both parents, but anyway, spontaneous mutations are possible. So the protein that's affected by the genetic disorder is the dystrophin protein. It affects skeletal and cardiac muscle. As you know, that will be a problem. Muscle damage and inflammation happen, and you get progressive muscle weakness. The clinical progression is generally ages 1 to 6 is when the first signs appear. The early sign is a waddling gait. The figure here is Gower sign, where instead of standing up from the floor, just jumping up from the floor like kids normally do, they use their hands on their thighs to stand, and that is because their legs are weak. And pseudo hypertrophy of the calves, so it's not real hypertrophy where the muscle cells are getting bigger, but the calves are enlarged because muscle cells are being replaced by fat cells and irretrievably replaced. By the time someone with Duchenne muscular dystrophy gets into their teens, they usually need to use a wheelchair for mobility 100% of the time. Ages 16 to 35, death from respiratory cardiac failure. So the mortality age has increased a lot in the last 20 to 30 years, but still it's a very early death. That's not good. Lots of secondary complications, and the cardiac and respiratory complications are kind of the most serious. So for physical therapy for Duchenne muscular dystrophy, we really want to maintain function and quality of life. Bed rest and immobility are harmful. They accelerate muscle loss and contractures and cardiorespiratory problems. So we're working on range of motion exercises to maintain joint mobility, gentle strengthening to preserve existing strength, cardiovascular activity to the patient's tolerance, respiratory training, posture to orthotics and bracing to support posture and mobility, assistive devices, and wheelchair training preparation for the future and wheelchair skills. So it's we're going to really try to improve function quality of life as much as we can. So medical management, the family and caregivers are going to get education on transfer techniques. Often physical therapy is providing that. Proper positioning, mobility expectations, home exercise programs. It's diagnosed with a muscle biopsy where they take a muscle sample and look at it underneath the microscope, and genetic testing. Sometimes glucocorticoids like prednisone are used to reduce the inflammation. There's this new treatment that's emerging. It's called exon skipping therapy, and what it is it's a genetic treatment that causes it's something that it causes the. I had to look this up, and it was really interesting. As the DNA is being encoded into RNA, it skips the defective gene. So I just think that's really interesting. So more to come on that. See, like I said, there's I have hope for a lot of these genetic diseases. So fund research. Funding research is so important. Off soapbox. So, for clinical applications for genetic disorders, understanding genetics helps us anticipate the disease progression, understand family risk patterns, provide appropriate education to family and caregivers and patients, plan long-term interventions, and recognize when referrals are needed. Remember the difference. Genetic doesn't mean congenital. There are two different things. With genetic disorders, inheritance patterns determine the risk. Some conditions appear later in life, like Marfan's and polycystic kidney disease. So, family history is crucial. If you know that someone in your family had a certain thing, you might be more prone to that. So, for PT, for Marfan, we're definitely going to monitor for cardiac symptoms and avoid high intensity activities because of the connective tissue issues. For cystic fibrosis, we're focusing on airway clearance and endurance. For Duchenne muscular dystrophy, we're trying to maintain mobility, prevent contractures, and avoid bed rest. So, red flags, cardiac symptoms, that's a red flag for everybody. But Marfan, you got to really watch for it. Rapid respiratory decline in cystic fibrosis and Duchenne muscular dystrophy, and loss of function beyond the expected progression. So we need to know the expected progression so we can see if they're losing function sooner than we expected.